Our bones are more than just rigid structures that give us our shape. They are living tissues, constantly growing and changing throughout our lives. This dynamic process, known as bone formation or ossification, is essential for our growth, development, and overall health. Imagine a construction crew meticulously building a skyscraper. That's similar to how our bodies create bones, laying down minerals and tissues in a precise and coordinated manner. There are two main types of bone formation: intramembranous ossification and endochondral ossification. Both processes contribute to building our skeleton, but they do so through different mechanisms. In intramembranous ossification, bones develop directly from a membrane-like structure. On the other hand, endochondral ossification involves a cartilage model that is gradually replaced by bone. Understanding these processes provides valuable insights into skeletal development, bone health, and the treatment of bone-related disorders. Intramembranous ossification is like building a brick wall directly, brick by brick. In this process, bone tissue forms directly within a layer of connective tissue called mesenchyme. Imagine mesenchymal cells as tiny construction workers that receive signals to transform into bone-building cells called osteoblasts. These busy osteoblasts then get to work secreting a specialized matrix that serves as the foundation for bone tissue. This process primarily occurs during fetal development, forming flat bones like the skull, clavicles, and some facial bones. Think of the skull protecting the developing brain like a helmet formed directly from this intricate process. Intramembranous ossification begins with the clustering of mesenchymal cells at specific locations called ossification centers. These centers act as starting points from which bone formation radiates outward. In the intricate dance of intramembranous ossification, various cell types play crucial roles. Mesenchymal stem cells, like versatile actors, have the remarkable ability to differentiate into diverse cell types, including osteoblasts, the master bone builders. Osteoblasts, resembling skilled masons, synthesize and deposit osteoid, the organic component of bone matrix, rich in collagen and other proteins. As bone matrix accumulates, some osteoblasts become trapped within their own creations, differentiating into osteocytes, the mature bone cells residing in lacunae, small spaces within the bone matrix. Osteocytes, like vigilant guards, monitor and maintain bone integrity, communicating with other bone cells through a network of tiny canals called canaliculi. Meanwhile, on the bone surface, osteoclasts, giant cells with multiple nuclei, act as bone sculptors, resorbing and remodeling bone tissue. Endochondral ossification takes a different approach, akin to creating a detailed ice sculpture that is then replaced by something more permanent. In this process, a cartilage model serves as a template for bone development. This process is responsible for forming most of the bones in our body, including long bones like the femur and humerus. Picture a cartilaginous framework like a blueprint guiding bone formation. Endochondral ossification begins with the formation of a hyaline cartilage model surrounded by a membrane called the perichondrium. Blood vessels then invade the perichondrium, transforming it into the periosteum, a membrane essential for bone growth and repair. This vascular invasion marks a critical turning point, initiating the transformation of cartilage into bone. Section 5. Stages of transformation, endochondral ossification unfolded. Endochondral ossification is a multi-step process, much like a carefully choreographed dance. First, a primary ossification center forms in the diaphysis, the shaft of the cartilage model. Here, chondrocytes, the cartilage cells, enlarge and die, creating spaces for blood vessels to invade. These invading blood vessels bring in osteoblasts, which begin depositing bone matrix on the remnants of the cartilage template. As the primary ossification center expands, secondary ossification centers appear in the epiphyses, the ends of the bone. These centers follow a similar process, gradually replacing cartilage with bone tissue. However, a region of cartilage, the epiphyseal plate, persists between the diaphysis and each epiphysis. This plate, also known as the growth plate, is where bone lengthening occurs during childhood and adolescence. Section 6, comparing the two similarities and differences. While intramembranous and endochondral ossification are distinct processes, they share some similarities. Both processes involve the differentiation of mesenchymal cells into bone-forming cells, highlighting the remarkable plasticity of these stem cells. Additionally, both processes contribute to the formation of the skeletal system, although through different mechanisms and in different locations. The key difference lies in the presence or absence of a cartilage template. Intramembranous ossification occurs directly within mesenchymal tissue without any cartilage intermediary. In contrast, endochondral ossification relies on a pre-existing cartilage model that is gradually replaced by bone tissue. This fundamental difference explains why intramembranous ossification primarily forms flat bones, while endochondral ossification forms most of the long bones in the body. Section 7. Clinical significance when bone formation goes awry. Understanding bone formation is crucial for diagnosing and treating various skeletal disorders. For instance, achondroplasia, the most common form of dwarfism, results from mutations in genes involved in endochondral ossification, particularly affecting bone growth at the growth plates. Craniosynostosis, on the other hand, involves premature closure of cranial sutures, the fibrous joints between skull bones, disrupting normal skull development. Fracture healing, a remarkable example of bone's regenerative capacity, recapitulates some aspects of both intramembranous and endochondral ossification. Initially, a soft callus resembling intramembranous bone formation stabilizes the fracture site. This soft callus is then gradually replaced by a hard callus, reminiscent of endochondral ossification, ultimately restoring bone integrity. Section 8. Conclusion. The enduring significance of bone formation. Bone formation is a fundamental biological process, essential for our growth, development, and overall well-being. From the intricate dance of intramembranous ossification, building bones directly from mesenchymal tissue, to the remarkable transformation of cartilage into bone in endochondral ossification, these processes underscore the dynamic nature of our skeletal system. Understanding the complexities of bone formation provides valuable insights into the pathogenesis of skeletal disorders, guiding the development of novel therapeutic strategies for conditions ranging from bone fractures to genetic skeletal dysplasias. As we delve deeper into the molecular mechanisms governing these processes, we gain a greater appreciation for the exquisite choreography of bone formation, a testament to the remarkable capabilities of our bodies. Section 9 references Marieb, E.N. and Hoen, K. 2019. Human Anatomy and Physiology, 11th ed. Pearson Education, Tortora G.J. and Derrickson B. 2017. Principles of Anatomy and Physiology, 15th ed. John Wiley and Sons, OpenStax, 2013. Anatomy and Physiology, OpenStax, CNX.